is 2, and I'm a cancer geneticist and a bioinformatician in the Department of Medicine at Monash Health. And I'm the convener of the MPCCC Tech Talk series. To begin, I would like to pay our respects to the traditional custodians of the lands from which we join together today, and acknowledge their elders past, present, and emerging, as well as any members of the Aboriginal and Torres Strait Islander community who may be among us. The MPCCC Tech Talks aim to provide an informal and interactive forum for our professional research community to discover and discuss the latest cancer research technologies and how they can be applied in a practical way to support our research. The Tech Talk format offers plenty of time for Q&A, and we really do encourage all of you to tell us about your experiences, key learnings and tips and tricks, as well as any potential challenges and limitations in the work you do. So today, I'm very pleased to welcome Tara Mack, dialing in all the way from Nashville, Tennessee. Thank you, Tara, for being with us. Tara is a human genetics PhD candidate at Vanderbilt University. Under the primary mentorship of Dr. Alexander Bick, her PhD work aims to understand progression rate of clonal hematopoiesis of independent potential, or CHIP. And more broadly, Tara is interested in the genetics of complex disease and genetic modifications and gene by environment interactions. So Tara is going to give us a presentation about identifying driver mutations of a CHIP in the blood. And so for the presentation, could I please ask everyone to please mute your microphones and turn off your video to minimize disruptions. So thank you, Tara, again, for being with us. I know it's late, so we should get started. Over to you.
Awesome. Yeah, thank you again so much for having me. I'm really excited to talk to you today about my work. And so I know, this word of sort of clonal hematopoiesis of indeterminate potential can be quite a mouthful. And so I'm going to kind of break this down for everyone. But so we're kind of talking today about just identifying mutations that are sort of cancer associated in our blood. And so to really understand this project, I think it's first important to understand the general idea of this clonal expansion phenotype. And so it starts with the assumption, that when you're born, all of the cells in your body have essentially the same genetic material, so the same DNA across all of the cells in your body. But as you age, that changes. And so as you age, we all accumulate mutations at a pretty regular rate over time across all the cells in our body. And most of the time, these mutations are neutral. They don't give that cell a growth advantage or disadvantage. They're sort of just along for the ride. And so we kind of term these passenger mutations. However, maybe a little later on in life, you might acquire a mutation that's different. And this is what we call a driver mutation. And it's different because it does confer a selective advantage or a growth advantage to that cell, which then leads to this idea of clonal expansion, where once that cell has that growth advantage, it begins to sort of rapidly expand and begin to take over a larger and larger portion of the cells in the body. And so we kind of term this a clone. And so you kind of can think of clonal expansion as a phenotype as sort of this idea of a yellow light. And so it tends to be an intermediate state between what you would call a normal cell and sort of a disease state, where you see the presence of this mutation in a subpopulation of cells in the body, but you don't yet see sort of any clinical consequences of those mutations. And so it can kind of serve as a warning signal that something may be coming in the future. And so specifically in our lab, we're really interested in understanding this phenomenon of clonal expansion, but in the blood specifically. And so this means that we're looking at these driver mutations that are acquired in our blood cell precursors, or our hematopoietic stem cells. And so when you get a driver mutation in your hematopoietic stem cells, this leads to this clone of dysregulated growing blood cells in your peripheral blood. And blood is a really interesting tissue to study because it's pretty accessible, right? So it's a lot harder to take, a slice of your brain or your heart and see these clones growing, but you can get someone's blood pretty easily. And so it's a really great tissue to study this idea of clonal expansion. And so when we say this sort of mouthful term of this clonal hematopoiesis of indeterminate potential, or CHIP, we're really just talking about what I just showed you. So this idea of you have this acquired driver mutation that's associated with blood cancer. So this is a specific list of blood cancer associated mutations that are present in at least 4% of your blood cells. So you have that driver mutation present, but you don't see any clinical consequences yet. So you have normal blood counts, you don't see anything like cytopenia, and you also have no clinical or pathological evidence for blood cancer. So like I said, sort of that yellow light phase of the mutation. And the reason we're interested in CHIP in general is because we know it infers increased risk of morbidity and mortality. And so individuals with CHIP are more likely to progress to blood cancer. They're also more likely to die from lots of different causes. And even when they have blood cancer, more likely to die from blood cancer. But what we've noticed in recent years is that there are more and more organ systems that are being implicated with CHIP. And so we've seen associations with heart disease, we've seen associations with kidney disease, COPD, lots of different body systems. You can sort of think that your blood is kind of flowing through your body and interacting with lots of different systems. And so dysregulation in your blood can impact a lot of different things. And so because of all these outcomes, we're really interested in understanding CHIP and understanding how it plays into disease risk. And we know that the prevalence of CHIP increases with age, which makes sense if you sort of think about this idea of you're accumulating mutations at a regular rate over time. The older you get, the more you're going to have, and the more likely one of them is going to make a difference. And so by the time you see people around 70 years old, around 10% of people have CHIP, and that increases exponentially as you age. And so when we're talking about CHIP, we tend to be focusing more on sort of an older adult population where we're really seeing this phenomenon. And one last really interesting thing is, like I said before, so these CHIP mutations are in a specific list of leukemia-associated genes that are sort of previously established to be blood cancer causing. And so this is a specific set of genes and a specific set of variants on those genes. However, it's interesting that you don't see sort of an equal frequency of these mutations across all the possible genes. So there's 74 possible genes. However, 2/3 of people with CHIP tend to have either a DNMT3A or a TET2 mutation, and the ones sort of listed here are the most common genes. And so you tend to see that most individuals have mutations on these genes, and that there's sort of a long tail of sort of less frequently occurring genes, mutations. And CHIP is something that we can detect in our peripheral blood. And so this is, again, when you're thinking about CHIP, you're thinking this is sort of a mutation that's present in a subset of the blood cells. And so you tend to see sort of a fraction of the blood cells coming back with that mutation and a fraction without that mutation. So this is what we call sort of our somatic mutations. So it's something we can detect in our peripheral blood based on our sequencing polls. And it tends to be detected in one of two ways. And so we can do this through whole genome or exome sequencing, which tends to be, this large scale sequencing of sort of your entire genome. And that's typically repurposed. So typically when you detect CHIP using these methods, it's something that you already had this data and now you're interested in studying CHIP. So this tends to be, again, more low coverage. So not as many calls are happening, but it is typically available in really large cohorts. And so that tends to be a common way to detect CHIP. But recently, as CHIP has become more interesting to the scientific community, there's also been a lot of sort of targeted panels that have been established to specifically identify CHIP. And so you tend to see those in sort of smaller cohorts or clinical diagnostics. And they're really cost effective and they can have really high sequencing depth, so they can be a lot more sensitive, but they're not really available in as large scale of a way as our whole genome and exome sequencing data. And so today I'm really going to try and focus a little bit on sort of the whole genome exome sequencing part of this, because like I said, this data is really increasingly widely available as sequencing gets cheaper. And it's something that is really important because this is really large cohorts that we're able to study CHIP in, but we also need to make sure that we're actually calling CHIP accurately. And it's something that we can do consistently across cohorts. And so kind of the effort I'm going to be talking about today is our work to sort of accurately and consistently identify CHIP across large scale cohorts. And I'm really excited to share this work with you. We have a sort of adapted this generalizable framework that we think will work for chip identification in large-scale cohorts. So this would work for sort of any data set where you had whole genome data. And I want to kind of show you guys, so this is the paper that I'm going to be talking about today and where this work comes from. And I want to definitely shout out the lead author, Caitlin Blashhart, who is the very much chip calling guru of our lab and has been really great at kind of streamlining this whole process. So I'm going to be kind of talking about a lot of her efforts today and efforts we've worked on together. But if you're interested in sort of the nitty-gritty details of all of this, please check out this paper because it's a really, really great resource if you're interested in doing chip calling. And so I'm going to kind of walk through just some general steps of this framework and how we think it can be applied to chip calling. And so again, sort of step one, an obvious step, is that if we want to identify chip, we first need to just identify our somatic mutations, which again are those acquired mutations as you age. And so again, if you think back to my graphic at the very beginning, when we're talking about clonal expansion, right, you're going to be identifying these red lines, right, these mutations that are acquired as you age. And those are what we're considering our somatic mutations. And so sort of in our pipeline, it gets a little complicated, but I'll give you the breakdown here. So essentially, we're going to be using a somatic mutation caller. So this is just a pipeline that there's lots of widely available callers that you can use. Mutec2 tends to be the one that we use the most frequency, that chip field uses most frequently. And all this does is it takes your sequencing files and it identifies somatic mutations that are present across the sequencing calls. And so you get a list of somatic mutations that are identified. And we tend to limit this search to our established list of chip variants. So we'll say, you know, we only want an output from these 74 genes and these specific variants. So we don't, you know, waste resources calling on the entire genome. And yeah, once we have this list of variants, this gives us a baseline to start off of just mutations, somatic mutations that are potentially chip variants that we can kind of begin with. And so that's kind of the first step that you would go through. And then we can kind of get into basic filtering. And so this part of the pipeline is a really good step for something that would be sort of applicable to really anyone doing this framework. And so this would work in basically any cohort that you were doing and just some basic stringent filtering that you want to do. And sort of the purpose of this is to make sure that you have high quality sequencing calls, right? So to be confident in a chip call, you want to make sure that you're confident that it was sequenced correctly and that it's actually a real variant that's present. And so I'm going to kind of show you. So here are some kind of red flags that would make me think, you know, this might not be a chip variant, right? And so if there aren't enough sequencing reads at that site, and so typically we use a threshold of at least 20 reads in general at that site. So if it's not adequately sequenced, we tend to not use that site. Importantly, if the alternative allele, so the mutation itself, is only sequenced only shows up a couple of times, that tends to be a red flag. And so we want to have at least 3 calls of that alternative allele to be confident that it's a real chip call. And then again, we want consistent sequencing reads in both directions to be confident that that's a real call. And lastly, because of the sort of the chip clinical definition, meaning that 2% VAF or that 4% of the peripheral blood cells, we tend to also filter here to make sure that our clones are at least 2% to kind of accurately call chip. So like I said, this step is really, really important and it eliminates a lot of these variants. And we want to make sure that we have high quality and reliable calls. And so these are general steps that I think pretty much anyone that calls chip will use. But sort of the next part of the pipeline is where I think our lab has really spent some time developing a really good method that really takes into account more factors than just these basic sequencing filters. And so again, the next step is sort of our population scale filtering. And so the idea here is that we want to make sure that the filtering can be adapted for the population or the cohort at hand, because every cohort is going to have different artifacts that show up depending on how the sequencing was done. There's going to be different germline variants that might sneak in. So there's going to be a lot of variation that you need to filter out. And so at this step of the pipeline, you have some real chip calls, right? But you also still have a lot of things that may still be present. You may have sequencing artifacts, you may have germline variants, you may have sort of those neutral passenger mutations that remain. And you, again, want to make sure that you're not leaving those in because that can really mess with your outcomes. And so the paper goes into a lot of detail on this and how you would eliminate each of these specifically. So I don't want to get too in the weeds here, but I can give you a general idea of what we tend to look for. So we know that CHIP is associated with age, right? And that's a well-established association. And so we know if we identify a variant and that variant is not associated with age, then that's probably not a CHIP variant, right? Like if it's not more common in older individuals, it's probably not a CHIP variant. And so some of the things we look for, right? If we see SNPs that aren't associated with age, we think, okay, this is a common variant, but it's not associated with age, so it's maybe a sequencing artifact. We also look for associations with known CHIP loci, so things that we know predispose individuals to CHIP. And so if we see association there, that is a good sign that it's a real CHIP variant, as well as we perform binomial tests. Can tell us whether something is a germline variant. And so binomial tests will just tell us whether it occurs at sort of that 50-50 threshold, which would be a heterozygous variant. But if it differs from that, that is more likely a CHIP variant. And so again, there's a lot of detail in the paper, and I don't want to bore everyone with lots of details, but there's a lot of really important filters in this step where you can go through and identify a lot of these artifacts. And in the paper, we have lots of supplementary tables where we've listed really common artifacts in all of these categories that you can look for in your data. And we did a lot of tests to make sure that all the variants we were leaving in fulfilled the criteria that we were expecting to be real CHIP calls. And so this step should really eliminate any of your false positives that you might have left in the data. And so I want to show you an example again of something that was really interesting in the paper, where this specific gene ASXL1 is one of our CHIP genes. And the mutations in ASXL1 were particularly likely to be sequencing artifacts, which is something that we noticed when we were doing this data. And so as you can see, like I said before, we wanted to test for the association with age for each of these variants, right? Because if CHIP is an age-related disease, we expect that the variants will also be associated with age. And so as you can see, these variants in the red here weren't associated with age, and the ones in the blue were. And so you can kind of focus in on just this top box here. You can see that when we kept just the blue variants, we saw that our variants were associated with increased risk of cancer, increased risk of death, and association with smoking history. And all of those things are highly associated with CHIP. However, we don't see this for these variants that are removed. We don't see them associated with any of these outcomes. And so this is a really good sign that when we're using these outcomes and these age associations as predictors for real CHIP variants, that we're really eliminating the noise and keeping the real signal in this step. And sort of the very kind of last step, and I know this is getting a little in the weeds, but I think it's really important, is this idea that because we're going to be using so many diverse cohorts, and there's going to be a lot of sort of health data linked in these cohorts, there's a lot of ways that you can further optimize and identify CHIP accurately in your data set. And I'm going to show you guys one of the ways that we did this in our analysis. So we use this framework to identify CHIP in both the UK Biobank and the All of Us data set. And doing that, we noticed some optimization steps that would be really cool to implement. And so one of the things we looked at was sort of our minimum allele depth for the mutation. So sort of how many times that mutation showed up in the sequencing reads. So we wanted to make sure that that threshold was a solid threshold. And so to do this, we tested the association with age for different sort of cutoffs for our minimum allele depth, right? So the one I showed you at the beginning, we did a three minimum allele depth cutoff. We also tested 4, 5, 6, so different thresholds. And we saw that as we increased that threshold and made it a little bit more stringent, we saw an increased association with age, which tells us that we're getting closer to real CHIP calls. And in fact, when we did some simulated misclassification experiments, we also saw that when we just used a minimum allele depth of 3, there were about 50% of those calls were estimated to be not real CHIP calls. And so it's really, really important that as we do these associations, we realize that this cutoff of 5 actually seemed to be a better cutoff here for identifying real CHIP. And this again showed up when we tested our sort of locus that we knew was associated with CHIP. we saw again that this 5 to be the optimal setting here. And so this is just one example of a way that we've tested to make sure that our chip variants are accurately called. And I just want to give you an example of why this matters. It might seem really in the weeds. You might say, why does this minimum threshold really matter? And the reason it does is because when you look at the outcomes, it makes a really big difference when you change that threshold. And so here, if you look at this top gray box, you can see that when we included allele depth of greater than 3, you can see that the risk of death was much lower than when you made a higher stringent threshold here of 5, meaning that we were underestimating the risk of death when we were including those sort of false chip calls. And again, this also showed up here when we're looking at our incident myeloid cancer. And so what this is showing us is that when we're particularly stringent on our chip calls, we are better able to estimate our risk of disease and death in chip, which is really important because if we underestimate that risk, that can be really, really detrimental to our patients and to really understanding how chip interacts in the body. And so it's really important that when you're implementing this threshold, you're also taking the time to do a lot of these associations, especially if you have like sort of outcome data, things like death and cancer, and making sure you're really seeing the associations that you're expecting to see when you're looking at chip and knowing that you're using accurate calls. So I know this got a little complicated, but I'm really happy to kind of start a discussion and really talk more about all of these things. But in general, just wanted to emphasize that I think this is a really, really important framework that you can use to identify chip. And it is complicated because identifying chip is complicated. And it's something that we really need to have a more central plan on how we do this across lots of different cohorts and lots of different research groups. And so I think that this framework both provides a structural background, but also a lot of flexibility to really optimize based on the cohort at hand. So yeah, really happy to take any questions, any discussion about the framework and any thoughts that people have. This is just a tiny part of my PhD work. I also study chip in a lot of different ways. So if you're also just interested in chip or interested in chip outcomes, I'm also happy to discuss that as well.
Thank you very much, Tara. That was a really great presentation. And for many of us who don't know what chip is, it was really useful. I'd like to open up for questions now. So if you have a question, could you please raise your hand or just and turn on your video? It would just be nicer. Do we have questions? Of Philip has a question.
Hi, Tara. Thanks for that. That was a fantastic presentation. I was hoping to ask if you have any experience with detecting chip in other tissues like saliva and how well you go about doing that.
Yeah, that's a great question. So I don't have a lot of experience with that. I know that there are some efforts to sort of understand this idea of clonal expansion in different tissues. But I know that sort of we've well characterized it in blood just because of the accessibility of blood, but saliva is also really accessible. So that is really interesting thought that we could capture this phenomenon in a different tissue. But quite honestly, I'm not entirely sure on the efforts on that. I certainly haven't done it, but I'm sure that in the coming years, we'll be looking at clonal expansion across all kinds of different tissues. I know there are some thoughts about how that can happen in the brain and all kinds of different things. And so I really do think that understanding how these mutations accumulate over time across all parts of the body is really, really interesting and will be an emerging field in the coming years.
Thank you.
Do we have any other question, Melissa?
Thanks, Tu, and thanks, Tara, for a fantastic presentation. Something that hits to the interest of a lot of people online. So it was really timely that you were able to come and present this and discuss it with us. So I have a couple of questions, and I thought the first one might be a bit philosophical and high level, but it may correct a very big misconception I have about CHIP. So I was interested in the way that you described the association between CHIP and other diseases, which I guess is a really interesting body of work that's evolving quite quickly. And you seem to describe it as an interaction between the blood, the CHIP as in the context of the blood and circulating, and the other parts of the body, whereas I guess I'd been thinking about it more in the context of common exposures. I wonder, can you tell me more about how you're thinking around this? I mean, I have very little background in this, and maybe that's a naive engagement of mine to think about it as a commonality, but what can you tell me? What else is known? Why did you say that? You must have a reason for thinking it in that perspective.
Yeah, I think that the true answer is that I don't think anyone actually knows. I think that both of those things could be true. I think that it's one of those things that we need more studies to really parse out whether there's some causality specifically from CHIP causing these conditions, or that there are, like you said, epigenetic aging, all kinds of other things in common that could cause these conditions. And I know for blood cancer and heart disease, we have these causative studies, right? We know that inducing CHIP in mouse models leads to these conditions. But for a lot of these emerging conditions that we're truly in the last year starting to see studies on, we don't quite yet have that data. And so there are a lot of theories on how CHIP could influence. There's a lot of genes that have, important pathways in these different conditions. There's a lot of sort of overlap in our sort of GWAS studies and genetic studies. And so I guess I don't actually know is the answer. I in my head, it seems to me that because, blood is so important in so many different aspects of the body, that it makes sense that dysregulation in the blood would impact other organ systems. But it is totally possible that, that's not true. So I think that's something that we need more studies on, and we just don't have. Like, I think that even like the kidney data was truly just published. Like, all of this is brand new. And so I think that it's something that we'll hopefully learn more about in coming years. But yeah, definitely don't take my word as fact, because I definitely don't actually know for sure.
I certainly wasn't saying you were wrong. I just sort of, I think it is emerging, and you said something that was quite different to how I was thinking about it. So being more informed, I was really interested in knowing. And I guess the scenario could be that the answer is different in different contexts.
Yeah, or both, maybe it's a combination. Yeah. I think it's a really important question, right? I think it's something that we're all trying to understand. I certainly would love to know. I think it's fascinating.
Yeah, I think a lot of these questions are going to come from very large cohort studies where the measures that are required to answer the questions that we've just discussed are actually available in the size that's needed to address these sorts of things from a statistically powerful position.
Yeah. And like I said, truly just this idea of being able to accurately identify CHIP across these large scale cohorts is something that is so important, because until we can do that, we're not going to have consistent data on prevalence of CHIP, outcomes of CHIP, all of these things, until we have a curated set of calls that we're confident in. And lots of different studies, even big companies have published studies where, my lab would not consider those CHIP calls accurate CHIP calls. And so I think it's really important to get this data first so that we can then get to those questions, right? Once we have a real data set.
Yeah, no, I think we absolutely appreciate that. And actually, I think parts of your paper are stuck around the walls in some of the offices in our spaces just for this very reason.
That's great.
We realize how fundamental it is, and that we shouldn't be flippant about the way we interpret data that we're generating in this space. Tu, can I keep talking, or am I dominating? I wondered how, so in the context of doing these studies in cohorts, and in some of our cohorts, we know that people have had various sorts of life exposures and even medical treatments for things. I wondered if you could comment on what sort of things we should be very attentive to when we're doing these sorts of analyses. I mean, some of our cohorts are cancer related, and chemotherapy and so forth. It's forever what we're worried about how we interpret our data. Do you have much advice or experience in that context, in how to interpret this data?
Yeah. Like I said, also a really emerging question. I think that first and foremost, cancer is very important and that it shouldn't be included. So any type of blood cancer diagnosis unless we know for sure that the sample was taken before that occurred, we never want blood cancer to be included. So usually in all of our cohorts, any blood cancer diagnosis is just immediately not included. And we also know from a lot of work, so one of our colleague, Kelly Bolton, does a lot of work on sort of the downstream effects of chemotherapy and even just inducing CHIP. So even non-blood cancer sort of chemotherapy can even cause a higher mutation rate, which can lead to CHIP. So definitely chemotherapy is a huge confounder as well. And so I think that that's definitely something to consider. And typically we tend to just avoid cancer in our cohorts, but it is an emerging question, right? Because that kind of can skew your data set a little bit. But you just have to be really careful and also make sure you know sort of at what age the blood sample was taken and when these outcomes occurred. So sometimes different cohorts will have different levels of clarity on that. You know, sometimes it'll be really obvious to you that, you know, the blood sample was taken 20 years before this diagnosis, but sometimes it's not quite as clear depending on sort of the medical record. And so that's really important. And then even just in my own interest in my work, so my PhD specifically is looking at sort of within people that already have CHIP, what underlies differences in CHIP progression rate. And so understanding sort of the rate of clonal expansion. And so I do a lot of work looking at all kinds of outcomes. So things like even smoking and alcohol exposures, all kinds of comorbidities and how those play into sort of the CHIP question. And so I'm actually currently working on a lot of projects to try and understand what other confounders could be playing into CHIP progression. But as of right now, we typically just don't include individuals with cancer or with known chemotherapy treatments. But that, you know, that might change in the next few years, depending on, you know, these studies.
Yeah, I think it was smoking that I was particularly thinking about when I used the word exposures. I mean, it's.
Yeah, smoking is associated with increased like CHIP prevalence in general. And so, which I don't know, it doesn't necessarily mean that I would exclude it. I mean, I think it's interesting that you could see that, but I think like, you know, chemotherapy tends to be a bigger, it kind of skews our mutation data in general.
Yeah. Yeah, no, it's fascinating. And you bring up Kelly's name. We are hoping to actually have her present at a meeting nationally this year. So yeah, there is a lot of local interest in this, and we're really looking forward to having her amongst us to talk this further on, I think in August. So we're all looking forward to that.
That's awesome. Yeah, her work is fascinating. I think it's really, really cool.
Sorry, I'll step back up.
No, it's okay.
Thank you.
Philip, you have another question?
Yeah, I was hoping to ask Tara, if you've, while sequencing the blood for CHIP, if you've ever sequenced another tissue to rule out the germline, and if that sort of added anything in improving the identification of CHIP when compared with germline variants?
So just sort of making sure that we're only identifying somatic mutations and not germline variants.
Yeah, because I know they do that often in clinic. So.
Yeah, no, that's an interesting question. I think it's it has been done, not on a very large scale, because we tend to only have blood samples on really large cohorts for the most part, at least the ones that I tend to be using for CHIP identification. But typically how we avoid like germline calls is through a lot of these filtering steps. So we make sure that we're seeing variant allele frequency that is not 50% or really close to 50%, because that would indicate that it's probably a germline variant. So we're tending to see a VAF of around, you know, anywhere from 2% to maybe 20-30% tends to be where we make those CHIP calls. And so typically if you're seeing that variation, that is a really strong sign that that's a somatic mutation and that it's not a germline variant, because you wouldn't really see that distribution, that allele frequency difference there. And so that tends to be sort of how we differentiate that. But I think it would be interesting to look at other tissues as well. But as far as I know, at least I don't think our lab has done that specifically.
Right, thanks for that.
So in our panel sequencing, for instance, we go down to 25% VAF for calling a variant germline. Do you think we should not do that if it's in TP53, for instance? If we have a P53 variant around 25-30%, how do we know then if it's germline or if it's CHIP?
Yeah, I think that the answer is you don't ever fully know for sure, except that you tend to get, so I think the way we typically know is because we have reoccurring variants that we see those associations with age or the associations with chip known variants, and we say, okay, this is probably a chip variant. And then there's other variants that tend to pass binomial tests, and we say, oh, these are probably germline variants. And we tend to sort of have these curated lists of variants that we're pretty sure fall into one or the other because we see them frequently. But I do think there is a case where you could have a variant that's present, or like a really large chip clone that looks like it's a germline variant. You know, you're never going to fully know for sure. But I think the other steps are how you differentiate that. So it's not just the VAF, but you're also looking for that age association, right? So a germline variant shouldn't be associated with age. It should be random, right? And so when you see that age association, you see that association with chip loci, you see that association with increased cancer and death and all those things, then those tend to be chip variants. And so when you get into the weeds there, that tends to be how we differentiate it, but it's never perfect. There's always ones where we're not quite sure, and it tends to be, we do this a lot. We have curated lists of things we see often, and there are fewer and fewer variants that tend to fall in that gray area as we get more experience doing this.
Do we have any more questions?
Yeah, I have a question regarding the filtering that you did. So like for allele depths, like sometimes you don't really get equal coverage across like every position. Some like positions you get very high allele depths and some very low, and that will act like for those with low allele depths, the VAFs can easily change based on just like a couple of reads. And like, how would you be able to take into account of that?
Yeah, that's a great question. I think generally the way we do that is because we tend to have a total allele depth cover or filter first. So for any site to even be considered, there has to be at least a depth of 20 or greater. And so that means that when we have that minimum allele depth for the alternative allele, it tends to be at least sort of 3 out of 20. So there tends to be sort of a minimum cutoff of sequencing reads that have to come in for that site in general first. And then once we're confident with that threshold, then we add that alternative allele threshold on top of that. And so any site that doesn't have an adequate number of reads would be filtered out before we would even get to the alternative allele depth. Does that make sense?
Yeah, I'm also curious, like, how did you come to the like choice of minimum allele depth of 20?
Yeah, so this is also just kind of one of those things through, and the paper kind of goes into a little more detail about exactly how all these thresholds were established. So I would also recommend reading that. It probably has more than I can even remember, but I think part of it is just through sort of the availability of data, right? So whole genome sequencing tends to be like 30-40x on average. So you kind of have to factor in what you typically have in terms of sequencing coverage for whole genome data. And then also sort of studies that we've done and published data out there looking at sort of what typically tends to be an adequate coverage for calling a variant at this threshold. And so if you check out the paper in the methods section, it should really lay this out a little bit better. I'm trying to remember the exact details, but we did a lot of testing and creating this filtering criteria. And it's also something that people use for somatic mutation calling even outside of CHIP. That tends to be a threshold that is used in this context.
Thanks for the answer.
Yeah.
Oh, can we please have the link to the paper? Yes, we can. Hannah, could we? Because we have emails. Or do you have a link handy, Tara? Or we could circulate.
Yeah, I can get one. Hold on. I'd have to. I can
Yes,
here. Do you want me to go back to the slide that has the link on it here? You guys can, the QR code, if you want, should take you, or you can just search the name of the paper.
I can also send it around to everyone who's attended today.
That'd be great.
Can I ask another question, Tara? Are chip variants more likely to be of a certain type, like indels, or they can be single nucleotide substitutions as well?
Yeah, so when we're talking about CHIP, we're talking about SNVs. However, when you talk about so clonal hematopoiesis of indeterminate potential, that sort of IP part tends to mean that we're talking about these leukemia-associated single nucleotide changes. However, CH, or just clonal hematopoiesis in general, can refer to a lot of different types of changes. And so it's sort of an umbrella term. And so you can see both things like loss of X, loss of Y, so really big changes. You can also see mosaic chromosomal alterations. So you can see those kind of changes, which are a lot more structural and large variations, deletions, loss of heterozygosity. And so sort of clonal hematopoiesis in general is like an umbrella term that can refer to lots of different changes. When we're talking about CHIP specifically, this is where we're talking about these leukemia-associated driver genes, and we're talking about a single nucleotide change.
Right. Okay. And for a person to be like, if you have CHIP, do you just need one CHIP variant? Or
yeah.
Okay.
Great question. That's actually, I probably should put that up here too. But so most people that have CHIP have one clone. So they have one CHIP variant that reaches that 2% VAF threshold. However, there are people that have more than one, and we tend to see that across all the cohorts. So you'll have, I think it's usually around 70% of people, 70-75% of people will just have one clone, but then you will see multiple clones. And so I actually also do some work studying kind of clonal dynamics when you have multiple clones in the blood, which is really, really interesting. So that's a whole other field of study, but just understanding how, multiple clones might interact or, potentially compete for resources in the blood if you have multiple clones growing. So it's also really fascinating.
Oh, yeah. We must have you back. We'll have you back.
Yeah, got lots of projects going on.
Do we have any other questions for Tara?
I had a really quick one if we've got time. So it's probably my naivety about CHIP. Are you guys using mutate in matched normal mode, or are you using a panel of normals? How does the somatic call work if you don't have matched pairs? Do you just take the whole cohort as a normal for everyone?
Yeah, so the details, just in case I say something wrong, are in this paper, so I would double check me on this, but I know we use a panel of normals, and I know that there's some steps, and it should go into like exactly how that works. I'm trying to remember off the top of my head, but I know there's a panel of normals, and I know that we specifically, again, filter that to just our CHIP variants of interest, so that speeds up the process. But there should be a lot of data on that if you want to replicate. I'm just trying to like remember off the top of my head exactly how we set everything up.
No worries, thank you.
Mm-hmm.
Well, thanks, Jason. That was also one of my questions, but I thought I might be talking too much. So, all right. Well, thank you again very much, Sarah, for a fantastic presentation and answering all our questions.
Yeah.
I think if no one else, if there aren't any other questions, we might just stop here. Thank you everyone for attending, and our next tech talk is June 20th with Rebecca Engel, who will be talking about patient-derived organoids as a clinical model of colorectal cancer. So you can register for this by visiting the MPCCC website. Thank you everyone again for attending. Thank you, Tara. Have a good night. Thank you very much for staying up to give this presentation. We really all enjoyed it.
Yeah, thank you so much for having me, and feel free, if anyone has any further questions, to reach out to me on any of the methods that have been shared. So
we will probably reach out. Thank you.
Thank you very much.
Sounds good. Thanks so much.
Thank you.
Bye.